Good morning once again. This time around, we are dealing with the process of nitrogen fixation. Remember, during denitrification, nitrate in the soil gets reduced into different kind of nitrogen gases, including N2, which is lost from the soil into the atmosphere. Unfortunately, nitrogen fixation is a process that enables the plant to be able to use that N2 gas which is found in the atmosphere. Ordinarily, the nitrogen is triple covalently bonded to another nitrogen, and it is not easy to break these triple covalent bonds. Only few organisms are able to do that. Then they break the nitrogen from the other, and then after that has happened, that nitrogen can either be combined with hydrogen atoms to form ammonium, and in some instances nitrous oxide can be formed, but largely the end product of nitrogen fixation is the conversion of an N2 gas into a combined form of nitrogen, mostly ammonia, NH3. That is basically what happens in nitrogen fixation. Now, you can see on this slide, what we're interested in is biological nitrogen fixation. Forget about the lightning and the chemical issues, but the biological nitrogen fixation. As you can see on this slide, they are showing you nodules on the root of the plant on the right-hand side, wherein you have got bacteria which are able to fix nitrogen. These are the plants that form symbiotic association with these bacteria, accommodating them in their roots in structures called the root nodules. These bacteria in turn have got an enzyme called nitrogenase, and this enzyme has got the ability to convert the N2 gas into ammonia. Only microorganisms can be able to do this, and high plants themselves on their own cannot be able to convert that N2 gas. So they team up with the bacteria to do this wonderful process. Here this slide is showing you about nitrogen fixation. Actually showing you that mostly the organisms that fix the nitrogen are the prokaryotes, the bacteria, and they're able to do that with the help of an enzyme called nitrogenase. You've got three types of bacteria, or different types of bacteria: cyanobacteria, rhizobium, bradyrhizobium. All these are microorganisms that are able to fix nitrogen. You can read this slide, it's very easy to understand. Now, there are three systems of nitrogen fixation, which I believe when you read them, you will be able to understand. I'm not going to go into them in great details. You've got the symbiotic nitrogen fixation, which is probably the most dominant, where the microorganisms and the plants form symbiotic association. Please read about that. You have got associative nitrogen fixation system, wherein you are going to have the bacteria living just next to the roots of the plant, benefiting by the roots releasing some root exudates, some compounds that the bacteria can use, but the bacteria does not live inside the tissue of the plants. And that is associative nitrogen fixation, as opposed to the symbiotic one. And largely you have got free living nitrogen fixation, where the microorganisms are living freely in the soil, not dependent on plants. they find ways of making their own, getting their own energy for fixing nitrogen fixation, and so on and so on. Now, this table is just showing you different types of these three different types of nitrogen fixations: the symbiosis, the associations, and the free living ones, highlighting the sources of energy for the nitrogen fixation, also saying a bit about the effectiveness of the system, and so forth and so forth. So you can also go through this table with nice illustrations just to help you to understand these three different systems. This slide is just showing you different associations, what type of a plant, what type of a bacteria forms an association with that particular plant. So this is all about examples of organisms that carry out nitrogen fixation, the relationship between the host plant and the nitrogen fixing microorganism or symbiote. So the slide is self-explanatory. I don't think you must have difficulties in understanding that. Thank you. I'm just going to go on with another slide, but I'm going to stop here so that the video clip can be able to, it's not going to be too big for me not be able to send it to you. I'll be back shortly. Bye. I'm back again, still on nitrogen fixation. This time around, we're going to pay specific focus on the formation of root nodules. You all know probably by now that legumes tends to form nodules. These are the structures that accommodate the bacteria Rhizobium. So there are different species of the bacteria in the genus Rhizobium. So basically, in this situation in which the Rhizobium is accommodated in the root of a plant in a nodule, there is an aspect which is called host specificity, which means a particular bacterium is going to infect one particular type of a plant species, but it can also happen that one type of a bacteria can affect more than one species of a plant, what is basically referred to as a host plant in this case. So basically, there are certain genes within the bacteria which determines specificity, which will direct which strain of a Rhizobium is going to infect which legume plant. Most of the plants that fix nitrogen are generally, I think most of them are legumes. So basically, in the formation of a nodule, remember, the bacteria will be living freely in the soil before it moves inside the root of a plant. So first you're going to have a bacterium living freely in the soil, and then it will come into contact with the roots of the plant. And then from there, a process that will result in the bacteria moving from the soil where it is freely moving into the root of the plant where it is accommodated in a structure which forms part and parcel of a root called the nodule. And normally the roots of the plants produce some chemicals called flavonoids and other types of chemicals which will attract the bacteria to the root and also helps the bacteria to determine whether the plant that is infecting is the right plant or not. So some Rhizobium can infect the root of the plant and not be able to form nodules. This could be referred to as ineffective strains of bacteria or Rhizobium and so on and so on. So the plant is going to secrete the flavonoids to attract the root node, the bacteria, and then the bacteria will move from the soil into the plant in a process that requires some kind of a communication, chemical communication between the roots of the plants and the bacteria itself. The rhizobium producing certain things, the roots producing certain chemical compounds, which sends a signal between the plant and the root, and other things will take place until such point that the bacteria moves into the root of the plant and is accommodated into the root of the plant. So I'm gonna skip this slide. Is the same as the previous one, just showing which host plant is infected by which strain of rhizobium or bradyrhizobium. These are just examples that you can look at. So when a nodule is formed, it is formed probably, as I've already mentioned, as a result of a series of communications taking place, chemical communications taking place between the root of the plant and the bacteria. I've already mentioned the role of the flavonoids. The bacteria produce certain things called the node factors. The node factors make it possible for the root hairs to be in such a way that they can be invadable by the bacteria, and those root hairs can form infection threads, and bacteria can be put in these infection threads, transported in these infection threads, all the way until they penetrate the main root, going through the different layers of the main roots until they are accommodated within the cells of the roots. And whilst they are dropped inside the cells of the root, they multiply and starts to form what is called the bacteroid. The plant, on the other hand, will be going through a lot of mitotic and meiotic divisions, making more cells on the root, eventually forming nodules, which will accommodate these bacteria. These are the bacteria that will later fix the nitrogen for the plant. So you can read these steps, the critical steps that are involved in the formation of the nodule. Read these steps, quite straightforward, probably 1 up to 6. This slide is also showing you how a nodule is formed, not so much different from the previous slide. Go through this. You can read, they are written in plain English. This is all about how a nodule is formed. Please go through this slide. And this slide is just probably here. I just want to highlight that, as I've already mentioned, the bacteria of a plant produces what is called the node factors. And when the node factors are produced by the bacteria, they elicit a number of responses, particularly on the part of the root of the plant. Deformation and plasma depolarization of root hairs and so on, go through all these responses. I can see 1, 2, 3, 4, 5 of them. The last one being inhibition of the reactive oxygen generating systems. This slide is just showing you in an illustrative manner what happens, the process that involves the formation of the root nodule. So you can try and understand this slide. Another slide also trying to show you how a root nodule is formed. It's basically just for illustration purposes. maybe they can help you to understand better how a root nodule is formed. This is where I'm going to stop. Finally, you can see root nodules on the root of the plant. This is basically what we mean by root nodules on the plant. Most of you have seen these kind of structures on different plants if you are the kind of people who like observing things. Thank you. Yes, the story goes on. The last time we ended on this slide, you can see this a slide showing the root nodules. I'm just quickly going to the next slide where you can see, depending on the plant species and different other issues, you have got two types of nodules that can be formed by a host plant. The nodule can either be determinate or indeterminate. So please go through this slide, read about the differences between determinate and indeterminate nodules. This slide is all about those differences. Thank you. I'm moving on to the next slide. Here, there's just a brief discussion about the nitrogenase enzyme. Remember, inside the root nodule, you have got the bacteria, rhizobium, and other types of bacteria. Within the bacteria, you have got the enzyme. This is the nitrogen fixing enzyme, and this enzyme is called nitrogenase enzyme. So what I can tell you about it is that it is an enzyme that is very sensitive to high levels of oxygen. High levels of oxygen renders this enzyme nitrogenase ineffective, and it cannot fix nitrogen under high concentrations of oxygen. And because of the sensitivity of the enzyme nitrogenase to oxygen, there are different strategies that have been adopted in nature that result in the enzyme nitrogenase being protected from high concentrations of oxygen. They are tabulated there. Go through them, 7 of them tabulated on this slide. Go through these mechanisms that are employed, or means that are employed by microorganisms to protect the nitrogenase enzyme from being inactivated by oxygen. So what does this enzyme do? The nitrogenase enzyme, as you can see, the first reaction, that is basically what it does. It converts the N2 gas into NH3, which is ammonia. This enzyme can also reduce acetylene gas into ethylene gas. You can see C2H2 being converted into or reduced into C2H4. It can also reduce hydrogen protons into a hydrogen gas, which is released. Obviously, it makes use of a lot of ATPs in the nitrogen fixation. Dealing with the biochemistry, I can just simply put it in this way. For the nitrogenase enzyme to function, it needs the energy. The energy is supplied in the form of the ATP. But remember, the source of that energy, the original source of that energy, is basically the carbohydrates of the host plant. So the leaves of the host plant are going to photosynthesize and form carbohydrates, and these carbohydrates will be delivered down the stem of the plant all the way to the roots of the plants where the nodules are. And within the nodules, this carbohydrate is going to be converted from the energy in the carbohydrate is going to be converted into energy in the form of the ATP. And this ATP is going to be used by the nitrogenase enzyme to fix the nitrogen. Like I said, the enzyme does not need high concentrations of oxygen. It is sensitive to a high concentration of oxygen, and therefore a lower level of oxygen is also required for the enzyme to be able to fix nitrogen. The N2 gas must be there. It is the substrate. If there's no N2 gas, then there won't be any nitrogen fixation, because nitrogen fixation is all about converting the N2 gas into NH3. NH3 being the product of the nitrogen fixation, N2 being the starting material. You will also need the energy, the electron donors, and so on and so on. So you can go through this slide, but I'm gonna try and explain the situation here. You can see you need the energy source, the carbohydrates. These carbohydrates will be produced by the leaves of the plant. You will need some electron carriers. Normally it's ferredoxine or FAD. You will need the enzyme itself. The enzyme nitrogenase is comprised of 2 units. One unit is called the ion protein unit, and the other unit is called the molybdenum ion protein units. These 2 units are linked together, and collectively they form the nitrogenase enzyme. So when you have got the energy, you can see I said the carbohydrates will be converted into the ATP. You can see here the carbohydrates coming from the plants being converted into ATP. There is that ATP. This ATP is going to be used by this nitrogenase enzyme here to fix that N2 gas to form the end product, which is NH3. Don't worry much about this C2H2. I told you this enzyme is also capable of converting acetylene gas into ethylene. So this is just another reaction that this enzyme is able to do. So same slide, I said you need the ATP there. You need the electron carriers. Here they are. No, the electron donors. Here they are. You need the electron carriers, the ferredoxine. You need the enzyme. The enzyme is comprised of an ion protein unit. As you can see, it is also comprised of a molybdenum ion protein unit. Both these 2 units together form the enzyme nitrogenase. So electrons will be donated by an electron donor, carried by electron carriers, and as they move from the one electron carrier to another, they will be received by the ion protein unit, which when it receives the electron is going to be oxidized, reduced. When it passes the electron over, is going to be oxidized. So here you have got oxidation reaction, reduction reaction. The electron will be passed over to the molybdenum ion protein unit. That's reduction. Passed over, that's basically oxidation. And eventually that electron will be received by the N2 gas here, which will be the final electron acceptor, and it will be reduced into NH2. Reduced into NH2. So basically that is in brief what nitrogen fixation is. The same slide dealing with one unit of the enzyme, another unit of the enzyme, nitrogenase 1, nitrogenase 2. These 2 units forms one enzyme here. So it's the same slide as the previous slide. Don't worry much about this slide. Ignore it. If we in class, I was gonna try and explain this to you, but don't even bother yourself to try and understand it for now. Don't worry about it. Thank you for now. Bye.